Hello everyone. You all know I'm Nilima. I'm currently doing PhD in Indian Institute of Science, Bangalore, and my topic broadly protein biochemistry, transcription, translation, regulation, and a few other things as well, like drug discovery and others. So today we'll talk about one assay called Bradford Assay. Now, why do we need to know about Bradford Assay? Uh, if we want to measure the concentration of any unknown protein, we have to do different kind of assays like Bradford assay. There are other method as well through which we can detect the protein concentration, but Bradford is mostly used assay in the laboratory. In any biochemical laboratory you can see, there people do Bradford assay to check the protein concentration. Now the first question comes that why do we need to measure the concentration of the protein? So the basic answer is, if you want to do any biochemical assays or any structural studies or anything, any kind of assays in the laboratory related to protein, you need to know the concentration of your purified protein. Now, let's say for structural studies, one protein in some paper it is mentioned that one protein got crystallized at the concentration of 15 mg per ml. Now you need to know how you can measure the concentration of your protein. Now let's say you want to reproduce that crystal for your study. Now whenever you want to set up trays for crystallization or any other biochemical assays, you need to know the protein concentration. What is the purified protein concentration in your hand? That is one question. So now Bradford assay is one of those assays through which we can detect our protein concentration. So today we'll talk about this Bradford assay, and after this video I will attach one video where I actually demonstrated in the lab how we do the Bradford assay. So now first we'll look at the theory part, like how Bradford assay work, and what is Bradford assay, how we can measure the concentration of your protein. Those parts, those questions, first we will try to solve, then we'll go to the experimental part, that the lab part. So Bradford is the name of the scientist who discovered this technique, this assay. Now it is nothing but one dye. So Bradford reagent is commercially available. It is available in the market. You can purchase it, and there are two kind of Bradford reagent available. One is non-diluted and one is diluted. If you purchase the non-diluted one, you have to dilute it first and then you have to use it for your assay. And if you don't want to dilute your reagent or don't want to do any kind of preliminary things before using Bradford reagent, you just can buy the diluted Bradford reagent and do the assay directly. Now one drawback, I will say, one drawback of this assay is that if your if you purify your protein in some buffer where some detergent is present, like Triton eggs or something, so some membrane proteins, like if you want to purify some membrane protein, you have to have used this kind of detergent molecule. So now if you have this kind of detergent molecule, then it's very difficult to actually solve what is the concentration of your protein, because these chemicals actually has effect in Bradford assay. But other than that, if you if your buffer containing NaCl, KCl, this kind of normal salt which actually stabilizes your protein in the buffer condition, there is no issue with that. But if your buffer contains detergent or some other molecule, then it is little bit difficult for you to actually demonstrate properly and solve the correct protein concentration. This is one drawback of Bradford, and one more drawback is that you can measure up to 1 mg per ml concentration of your protein. But there is a technique where we actually dilute our protein before do the Bradford. Then we can directly multiply that dilution factor with your concentration to get the final concentration. That also you can do. Now we will go to the theory part of the Bradford assay. So as I told, the Bradford is the name of the scientist who discovered this assay. Now Bradford reagent is nothing but one dye called Comasi brilliant blue. Actually, we will check the absorbance shift of this chemical, that is Comasi brilliant blue, in this assay. Now this Comasi brilliant blue actually can exist in three different forms. One is cationic, one is anionic, and one is neutral form. So now the cationic form is the blue in color, and anionic is the red in color. So normally, if you see Bradford reagent, you can see it's red in color. So normally, if we like whatever reagent we stored in 4 degrees Celsius, this is actually anionic form, the red one. And if at the neutral state also it can exist in the neutral state as well, that is green in color. Normally, if you see in your lab, you can see it's actually red in color, that is the cationic. But when it interacts with protein, it changes its color to red to blue. Now how this color change happen and all these things, that we will discuss. And I will also demonstrate in the experimental way, where I will show the change of color in the protein solution, where we actually can see the change of color. So now, as I told, there are three forms: cationic, anionic, and neutral. Cationic is red, anionic is blue, and neutral is green in color. Now, normally it is exist in cationic form, that is the red form. But whenever it interact with the protein, it convert itself into the anionic form, that is the blue form. And we can detect this blue form with the uh, 9 with the 595 nanometer of the of a light. So this is the wavelength of the light for the assay, which we use for the detection of this conversion of the Bradford reagent from the cationic, that is the red form, to the anionic, that is the blue form. Now how it actually occurs? Now if you see the structure of the Comasi brilliant blue, you can see there are different parts that can interact with the protein molecule. Now, what are the regions? Like, if Comasi brilliant blue wants to interact with the protein, there are particular side chains or some parts of the protein that actually interacts with the brilliant blue. So there are two functional group is there. One is carboxylic group and one is amino group. We all know in the amino acid and in the peptide chain, the protein has two functional group. One is carboxylic and one is amino acid. Now, this brilliant blue can interact with this carboxylic and amino group. Now, for the interaction with the carboxylic group, that dye actually interacts the van der Waals interaction. We all know this is the actually very less stable interaction, van der Waals interaction, and brilliant blue actually use these kind of interactions strategies for the interaction with the protein. And there is also amino group, but with the amino group, it interacts with the electrostatic interaction, that is the plus minus, that is positive negative interaction. Here there is no van der Waals interaction, only there is electrostatic interaction through which the dye interacts with the other part, that is the amino part of the amino group of the amino acid, that is the peptide chain. So as I have discussed, this is the main mechanism of Bradford reagent. So nothing but it's a dye, that is the Comasi brilliant blue, and it interacts with the protein and it converts the color of the protein solution. By only seeing the solution, you can say if there is protein or not. But for the quantitative assay, that is the measurement of the protein concentration, you need to measure the protein concentration with this wavelength of light, that is the 595 nanometer. From there we will get a absorbance value, let's say 0.122. Let's say this is the absorbance value we got. Now what we will do, so before this Bradford test of an unknown sample, we first make a standard curve. So what is standard curve? We first take 0 mg per ml protein to 1 mg per ml. Let's say 0, 0.2, 0.4, 0.6, 0.8 and 1. These are the mg per ml. So these are known concentration. Now what we will do, we will check the absorbance value of each of these concentration of the protein, and we will plot it. So it's like X and Y, and it will be like this. And after 1 mg per ml, it will be like saturated, because we can detect up to 1 mg per ml concentration of the protein. So now we will get like, let's say in case of 0 mg per ml, we got 0 absorbance, because there is no protein, there is no absorbance. Next 0.2, we got some absorbance value. In 0.4, we got some absorbance value. 0.6, we got something. 0.8, we got something. And 0.8 and then 1, we got something. And then it's saturated. Now, based on this plot, we have a standard curves. It's called standard curve, and we have a equation, that is standard curve equation. There will be X, there will be Y. Okay. Now, what we will do, we have this absorbance value. Let's say it's 0.122. Now you can see here 0.2 is here. So 0.122 will be somewhere like here 0.122, and from here, and this is mg per ml, and this is the absorbance. Now at 0.122 we can see what is the mg per ml, because this axis is the mg per ml, and if we put this absorbance value in that standard curve equation, we can calculate the mg per ml. If you don't know, if you have confusion, you can just let me know in the comment section. I can give an example, give one of my assay example where I detected the protein concentration from this absorbance value, because this I can get from the machine, from the Bradford, the computer which is connected to the Bradford machine. I will get this value there, but from there, how you get the mg per ml, that is the protein concentration, that is one task as well. So if you don't know how to calculate it, let me know in the comment section. I will give another video how can we calculate from absorbance to the mg per ml. That is one thing. And this is the theoretical part of the Bradford test. Next, with this video, I'll attach another video where I experimentally I'll show you how we can get this absorbance value. In my video, I got that video up to this level, the absorbance level. But if you have any confusion or doubt from here, how you can get the mg per ml, that is the protein concentration, just let me know. I'll show you how you can do that. From my assay, I have done the standard curve. I have done from this absorbance to mg per ml in my today experiment. It's just a 5-minute video, so it will be a 5-minute video. So just let me know. I'll put one other video as well. Stay tuned. After this I will put another video, that is the experimental part, and let me know if you have any confusion, any doubt. Just let me know in the comment section. I'll give you the solution. Thanks for watching.
Hello everyone, after a long time I am back in my YouTube channel where I will discuss about one experiment. So today we will do one experiment where we can detect the protein concentration. There are multiple methods for detecting protein concentration, but today we will do the Bradford experiment in which we will use Bradford reagent for the detection of protein. So stay tuned with me. In industry, there are two kinds of Bradford reagent is available. One is this one. This is the non-diluted Bradford. So, and in our lab, the diluted that is 1:5 ratio. The dilution ratio is 1:5, where the Bradford reagent is diluted into 1:5 ratio, and like 5x diluted Bradford, in which in this experiment I will use this diluted Bradford, because if I use this one I have to first dilute it and then use it, but this is already diluted and I made my standard curve of Bradford with this. So I will use the same standard curve for detecting my protein concentration directly from the absorbance. So for this experiment I will use this one. So first we will take 1 ml of Bradford reagent in each of the tubes. So this is my blank, and I want to check the concentration of 4 different protein. So for that I have total 5 tubes. So in each tube I will take 1 ml Bradford reagent. Now we will keep this in 4 degrees Celsius. Now with the blank I will add 20 microliter of the autoclaved molecule. Make sure your molecule is properly autoclaved and there is no contamination in it, otherwise you will get a false rating. So directly 20 microliter I will add in the blank. Done. And 18 microliter in the test sample, because my protein is already diluted, so sorry, my protein is already concentrated, so I will make sure that protein concentration should be less than 1 mg per ml in the test solution, because through Bradford we can detect up to 1 mg per ml protein concentration. So what I will do, I will dilute my protein into 10-fold and then add into the Bradford reagent. So first I am adding 18 microliter of the autoclaved molecule with the test vial, in the test vial. So I have 4 test vials, so I am adding 18 microliter in each. Done. Now I will add 2 microliter of my protein in each of the test vial. So now I have the concentrated protein, as I have already added 18 microliter of molecule. So when I will add my concentrated protein, it will dilute. So that is the logic. That's why we are diluting our protein in the Bradford and then measuring, because it can detect only 1 mg per ml up to 1 mg per ml protein. So that's why I first add 18 microliter and then I will add 2 microliter. So it's like 10-fold dilution. So when I will calculate that time, after measuring the concentration, after measuring the absorbance, after calculating everything, I'll multiply that concentration into with 10, so that I can get that the correct concentration of my protein. So now I'll add 2 microliter of the each protein in the test vial. make sure your pipette is good enough, so that it can like calibrate it properly. Otherwise it will take wrong amount, and you will get a wrong measurement. So make sure that. So first I will take RNA polymerase. So I am doing this because of one of my assays, so for that I need the concentration. So first I will add RNA polymerase. I'll shake it properly so that like with the reagent my protein is mixed well. Next, so with blank we will not add any protein, only 20 microliter of the molecule. Now sigma factor, then some accessory protein also we need for the transcription. So I am adding that accessory protein. Maybe you can see the color difference of to this vial. This is the blank and this is the protein added. This is more bluish black in color comparing to this. So you can check the color of these two vial and you can see there is protein. So now I need to check the concentration. So now we will do that. So for measurement, I need the protein sample, I need the cuvette. This is the cuvette. Make sure it's cleaned properly with alcohol and water. I need milky, I need alcohol, I need tissue paper, and for discarding the water, the worst water, I need 1 beaker. Now I am going to the central facility of the department. So there is a symposium going on, so people are there. I need to do this experiment, so I am here in the lab. So first we will go to the nanothrop and open the system. So here you can see protein Bradford. We will click it. Fine. So you can see here it is asking for a routine verification of the wavelength, and make sure the arm is down and click OK. So the arm is down. So normally we place one tissue paper like this, and keep it like this. But when we want to measure the concentration of the protein, so you need to remove this tissue paper and the arm should be down, and you have to click on OK. So see, performing routine wavelength verification. So it is doing now. So it is done. So now as we are using cuvette, you have to click on use cuvette. Click it and add. So then for blank we need to put the blank here. So we will up the arm, and in the cuvette I have already put the blank sample. So here you can see one triangle is there. You can see it should be like this. So this arrow is the path of the light. So this arrow, so you can see the arrow should be this side. So we will place it like this. Then we will move the arm down, and then we will click at blank. When the blank is done, the measure will be like green in color. So you can see here, see now it's green in color. So this plank is done. Now we will do one by one the measurement. Then I'll click the measure. Now it is measuring the concentration, like the absorbance. So here the system is telling to save my measurement, but I don't want to save it. I just take it as a photo and I'll calculate. So I'll cancel the saving. See, it is showing something minus 0.003, some absorbance, and now we will calculate accordingly based on this. So in this way we will, so this is the absorbance value. So this is the first sample, and for other sample as well I will calculate accordingly.